Thank you, Helena, and thank you to you and Michal for organizing this great meeting. Some of you may be old enough to remember the MHC evolution meetings that used to take place in the 90s and early 2000s, and maybe this is a revival of those meetings. They were really good. Anyway, today I'll tell you about our work on MHC class 2 genes in the Paleognathous birds, which you see here on the screen. They're the flightless birds, which many of you are probably familiar with, including the ostrich from Africa, the cassowary and emu from Australia, the kiwis from New Zealand, rheas from South America, and the tinamou, which is the small bird in the center, which is the only one of this group that can actually fly. And it can't do that very well. So there's been a lot of new genomes online, and some colleagues and I have summarized this explosion for you on this slide. Of birds, there's about 500 genomes now. Now, when I say genomes, of course, that's with a asterisk on it, because not all these genomes are high quality. And we know that to really get a handle on MHC genes and complex multi-gene families, we need to have as good a genome as we can, preferably with long-read sequencing. Now, the genomes that I'll be telling you about today are not, there was short-read sequencing, so they're sort of first generation. We think they're, better than many, but certainly there's lots of gaps. I will say, though, however, that I think manual curation and annotation can go some way towards helping us understand the MHC, even in these suboptimal genomes. And of course, all of us are thinking about long-read genomes now for our favorite species. Emily O'Connor and Helena and I and several others just were put together some information on the MHC, handy review in birds from a couple of years ago, and just outlining some of the advantages and disadvantages of different sorts of approaches, and also just noting this huge increase in next generation studies of MHC genes starting about 2010. Not all of them involving genomes, some of them involving sequencing of fragments. And just to introduce one of the themes I'll be talking about is this really nice study by Michal et al. I think Poitar was here yesterday, which we kind of presented in our review. And it's, although most bird genomes are incomplete, I think we're confident in asserting that there's lots of variation in gene number, especially among, well, more so probably among class 1 genes, but also among class 2 genes. And you can see, for example, on the bottom here that the suboccine birds and many groups of passerine birds seem to have lots more class 2 genes than other birds, although these numbers are obviously subject to revision. Now, the Paleognathous birds are important because they are the sister group to all other living birds. You can see them here on the bottom of the screen. And as the sister group to all other living birds, they hold a special importance for helping us understand the ancestral state of the MHC in birds. Reconstructing ancestral states is a fine art, and it's frankly very difficult to do, but certainly can lead us to certain conclusions and suggestions once we understand the MHC structure. So a few years ago, my lab sequenced a bunch of Paleognath genomes. We were interested not so much in the MHC, but in how they lost their flight. We even inherited we analyzed the genome of a little bush moa, which I am currently trying to write up. And among other things, we confirmed that the tinamous, the one group that could fly, is actually nested within the flightless species, which is a big surprise. And this suggests that there's been a lot of convergent loss of flight rather than an ancestral origin of flightlessness. Now, we found, and also another group recently reported, that these paleognath genomes are about 27% larger than those of other birds. And this likely impacts the MHC. So you can see the distribution of assembly sizes here. There is one long read genome from an emu. And an interesting twist to the story is that, surprisingly, that long read emu genome does not seem to have the homologue to chicken chromosome 16, whereas we find fragments of that microchromosome in our short read assemblies. So basically, Alison Cloutier, a postdoc in my lab, and I have just been sort of laboriously blasting away and trying to piece together the MHC. And you can see that we found multiple class 2 beta chain genes and alpha genes in various of these genomes. Each of these lines here is a different scaffold. So you can see that it's not assembled into a single scaffold, but we have some preliminary information. Now, surprisingly, in the little spotted kiwi, Apteryx oweni, we found as many as 6 class 2 beta chain genes. You can see 4 of them on this contig on the top, all within a distance of about 15 kb. And then on the bottom, on another contig, we have an additional 2. One possibility is that the high density of transposable elements in this region has contributed to duplication in this region. We don't know whether all of these genes are expressed. We suspect that at least these ones on the top are functional, although you know, we haven't really established that yet. They're not completely annotated, but they do have full-length exon 2 and 3. Now, what we found essentially is a gene order in the MHC, which seems to reflect the canonical extended class 2 order that is found in humans and other mammals. And so, for example, genes such as COL11A2, RXRB, and DAX appear on similar or nearby scaffolds of the MHC class 2B genes. And so this suggests that the paleognaths have a extended class 2 region, which is similar to that found in humans and other mammals. And if you line up the human MHC and the paleognaths and other birds, you come to a conclusion, which many of us have been suggesting for a long time, that the chicken is actually unusual for birds, that its gene order and its size in particular doesn't necessarily reflect the diversity found in birds. The paleognath MHCs in the class 2 regions we've been looking at are on the order of about 200 kilobases, so quite a bit larger than the entire chicken MHC. And then I'll just end with a few slides showing the evolutionary dynamics of this gene family. When we look across the paleognath exon 2 of these class 2 genes, this is the peptide binding region, of course, we find reasonable evidence of adaptive evolution. You can see the omega here values here on the y-axis. When we look just within the those 6 kiwi paralogs that I mentioned, we also find evidence of diversifying selection. And finally, we find pretty compelling evidence for gene conversion among these kiwi paralogs. Basically, different regions of the gene show strongly different trees, which are significantly conflicting. And I'll end there, and I'd be happy to take any questions. Of course, our next steps are to look at expression and to get some long read genomes so that we can really map these MHCs definitively. Thank you.